over to me. I am the host for this English session, moderator today, because yeah. So good evening, everyone, and welcome to this English session of the IFPH, International Forum for Promoting Homeopathy. Let us have a 1 minute silence prayer. Thereafter, we can start the session. Thank you. As you know, that International Forum for Promoting Homeopathy is being working since last, and now we shall be completing about 1600 days on 16th of the January. This is a wonderful journey for us, and we have traveled all these days with so many speakers, so many well-wishers and the participants like you all. Friends, unlike every day, we are having a new speakers, and they come here to make their presentations. We invite them, and today in this way we have our old and regular speakers, Dr. SURAJ Singh Bhadoria. I would like to introduce you SURAJ Singh Bhadoria as a faculty member from the Parul Institute of Homeopathy in Baroda. He has postgraduate degree and also the graduations from the Bharati Vidyapeeth, and then he has presented so many papers, and his articles and were published in the peer-reviewed journal. So, and he has been awarded also. So, without today, he is going to discuss with us the challenging aspects of the challenging phenomenon, things that is being felt during in the practice of the pharmacy practice. Because he is a teacher from the pharmacy, he will be speaking on the pharmacy and about the pharmacy. So over to you, Dr. Suraj. You can start your session now.
Am I audible, sir?
Yeah, you are audible now.
Okay, so first of all, I am very thankful for this platform to explore our knowledge in the great and the diversity and the spectrum. So let's I am going to sharing my PPT with you.
You can do it because you are the co-host now.
Okay, sir. Is it visible to all, sir?
Now it is coming up.
Now it is visible, sir?
Not yet, but it is coming up. It has started sharing now. Screen sharing is started. It is yet to be uploaded.
Yes, sir.
Yeah.
Now it is visible, sir?
Yeah, now it is visible.
Okay, so first of all, good evening to everyone. Today, it is not a topic. It is an important aspect as a homeopath, that what are the challenges in our homeopathic pharmacy. Okay, so first we will discuss in the challenges. Now, the first thing is identifying the drug substances. Clear? So, as we know, the homeopathic pharmacy, it is a branch of pharmacy. And the pharmacy is a branch of science which deals with the identification, selection, collection, preservation, preparation, and dispensing, the drug development, the drug proving of any material of drug substances. So basically, when we talk about the identification, so identification plays a major role. How we will identifying the drug substance by various sources. Because as we know, in pharmacy, there are different sources through which we prepare the medicine, we collect the drug substances, and we make a certain formulations. So challenges in the identification. So what are the challenges? The first thing is the historical background. We have to understand about the historical background, where it originated, what is the history behind that particular substance, and how it progressed in the field of pharmacy. So the history background plays a major role. The second thing is the taxonomical classification. Taxonomical classification will give you an idea about that particular classification of the drug substance or the medicinal substance. So the taxonomy makes the classification, it makes the categorization, it makes the nomenclature. So that will be the very challenging in the identification of drug substances. So first we have to know about the historical background, then the taxonomical classification, and the third one is the integration in the allied sciences. Previously, we always talk about homeopathy, homeopathic pharmacy. Now, after this new curriculum designed by the NCH, new CBDC program for both BHMS and the MD course, now there should be an integration. What is integration? There is a union and there is a coordination of homeopathy with some other sciences, like homeopathy with zoology, homeopathy with botany, homeopathy with the agricultural sciences, homeopathy with the veterinary sciences, homeopathy with the some other chemistry and the physics branch. So this is the integration. So this is one of the challenge while doing this identification, because we are the homeopath. We are not master and specialized in the light sciences. We know about the homeopathy, how the homeopathy works, how it forms. So by the integration of these allied sciences, we can make a strong union and the judgment to identify, to collect, and to prepare our medicine by the drug substance. So this is a very, very important point, and it is now included in our academic part also, in our syllabus part also. Previously, research was only done with the PG and the PhD students, but now the research also integrated with the bachelor students. So this is what? This is the integration. The last thing is the drug knowledge. You have to know about the drug knowledge. For example, if we talk about Apis mellifica, so we have to know about what is the preparation part, what is the active principle of that particular drug substance, so that it can show the therapeutic value also. Clear? Not only knowing about the drug as a source, but we have to also know about their compositional value. We have to also know about their chemical composition. So this is the drug knowledge. So these all criterias, and actually these are not criterias, these are the various parameters which included under the identification of drug substances. So if we know, and if we talk about this historical, taxonomical, integration, drug knowledge, so these all are the collaborative parameters. So when we make these parameters in a chain, in a sequence, so we are easily going to identify the drug substances. Clear? So this is the first challenge. If we know about taxonomy, if we know about their alkaloidal properties, so we can easily make the medicine from that particular source. And if we don't know about this particular parameter regarding to that particular source, so somewhere we are unable to make it as a evidence based, because now again homeopathy is considered as a evidence based medicine at international level also. So why? Because there are some lacunas not in homeopathy in our knowledge. Clear? Every branch having its speciality, the originality. But as from our side, we have to inculcate, we have to nourish our knowledge with the various ground, with the integration of multidisciplinary, so that we can make a holistic approach. So there should be also holistic approach in pharmacy, not only in prescribing medicine. Clear? Moving towards the next slide. Now, what are the challenges in the collection of drug substances?
Dr. Suraj, you unmute yourself. I have unmuted all. There was some sound loud voice coming. You unmute yourself and you start again. You
Now
just
it is
unmute.
audible?
Yeah, you are okay. You audible.
Okay. Okay. So previous slide, is it going to, like, we will all again discuss this. So in identification of drug substances, what are the challenges? In historical background, in taxonomy, in integration with the light sciences, and the drug knowledge. Okay, so these are the challenges. When we talk about historical background, so we have to know about the origin, we have to know about the history of that particular drug, of that particular source. Second thing, we have to know about the taxonomical classification, their species, their categorization, their groups, their division, genus. So these all are the taxonomical classification. Integration in the light sciences. It means the coordination of other branches like branch of physics, branch of chemistry, branch of veterinary, branch of agricultural sciences, branch of chemistry with the homeopathy. So when there is an integration, so we can easily make a holistic approach in identifying the drug substances. And this integration, it is also suggested by the NCH for both UG and the PG syllabus. So integration is a major criteria. It is a major parameter to make a evidence-based medicine. Drug knowledge, definitely. As a pharmacy people, we should always have a complete picture of drug. So we should know about their characteristic property. We should know about their alkaloidal property. We should know about their physical property. So the drug knowledge is related with the pharmacognostic study. Okay, so pharmacognosy is a branch of pharmacology. Clear? So these are the challenges while identifying the drug substances. Moving towards the next slide. Now, what are the challenges in collecting the drug substances? So as we know, while collecting the drug substances or while preparing the medicine, when we talk about that particular source, so there are certain parameters and there are certain factors which influencing on that. The first thing is the climatic condition, because as we know, different geographical area, different places having different climates. On the basis of that, that particular source, suppose if it is plant, suppose if it is vegetable, suppose if it is animal, suppose if it is mineral, so their alkaloidal property gets modified by this external environment. So the climatic condition plays a major role. We have to know about that in what climate that particular alkaloidal property or the therapeutic value of that particular drug increases. In which climatic condition the therapeutic value of that particular source or the drug material gets decreased. So these are the very important parameters or the factor influencing while collecting the drug substances. Rituals and worship. Previously, when we talk about Ayurveda, which is very oldest system of our Indian medicine, so in the previous era, people, when they are going to collect the drug substance, so first they always go for prayer in front of the source, in front of that particular plant. After this prayer, then there is a modulation, there is a modification, there is a synergistic action in the therapeutic value of that particular constituent or that particular chemical compound which is present in that particular drug or the source. Clear? But nowadays, this part is somewhere like 50-50. It is not 100%. So why? So we have to know about this particular concept also. What are the scientific and what are the philosophy behind this? So as it is like when we talk about homeopathic preparation, so we can also do like that. So when we do like that, so we can also make it as a more dynamization or more influencing in terms of therapeutic value. Okay, suppose if we have a nervousness, if we have anxiety, so we always want to go in a temple, in a church, in some different places. Why? Because something there is a positive vibes or the existing of energy. that will make you as a positive individual. Similarly, like that, when we talk about the drug, so when we do prayer in front of the plant or the any source, so that will make a connection in between two different forces. First is human being and second one is the that particular source. So that union make modulation in their therapeutic value. Clear? The third one is the active part use. Which part is going to use for making the medicine? So that is also a very challenging job, very important influencing factor while doing the collection. So suppose when we collect the roots or the rhizomes, so that should be an active part. It means they have all the alkaloidal property, they have all the, what we can say, the chemical composition through which we can go for the therapeutic value in that particular living entity. So the collection part plays a major role. Okay. Now moving towards the next slide. Now it is regarding with the preparations part. Now again, when we talk about homeopathic pharmacy, so we have old method and the new method, Hahnemannian concept and the new method. So in old method, we have 9 classes as per different nature of drug, mother tincture, mother solution, mother substances. And in the new methodology, we have maceration and the percolation. So what is maceration? Maceration is a process through which we are going to make it as a potentized by the juicy, gummy, mucilaginous drug substances. Whereas in the percolation, the drug substances are non-gummy, non-mucilaginous and the dried. So these are already written and these are already standard in our preparation. But what are the challenges? The challenges are when we do trituration, when we do succussion. So there should be a standard design. There should be a standard methodology for the time interval, for that process, for the mechanical inversion, and for the instrument part also. So that should be specifically designed, that should be specifically categorized. So we will make this procedure as a standard. Okay, so there are some other parameters, there are some other components which make a standard procedure for any pharmaceutical preparations, because pharmacy is definitely, it is more over the pharmaceutical preparations. The another thing is the drug vehicle ratio. The drug vehicle ratio, we always talk about the pharmacopoeia. I'm not against pharmacopoeia. We always have this background because these are the standard. Okay, so when we talk about 1:9 and 1:99 or 50 millisiemens, why not other ratios also? Clear? So when we prepare, suppose when we talk about lotion, okay, so suppose as per pharmacopoeia, lotion prepare in the category of 1:9 ratio. Suppose. So why not we prepare this lotion in all the ratio? 1:1, 1:2, 1:3, 4, 5, 6, 7, 8, 9. So when we do this all ratio from 1 to 10, then we can easily compare the results, the analysis, both qualitative and quantitatively on all the ratios. Clear? So this concept should be also included in our practice of doing this pharmaceutical preparation. So that will give you a different idea, that will give you an evidence based which ratio give you more effective results in terms of analysis, in terms of quantification, in terms of qualification. So these are very, very important. Not always talking about 1:9, 1:99 and 50 millisiemens. We should always have the multiple options. In modern pharmacy, they were like preparing a different ratios and they are going to compare it. So why not we people are doing like that? Clear? So our mentality should be in the broad spectrum. It should not like a very specific. Pharmacy is not specific. Pharmacy always flow in a field of sea. It is just like a sea. This is samudra. Clear? Segregation. What is segregation? As we know, our medicine having n number of alkaloids n number of active principles, more than 100 or maybe thousands. So by the process of chromatography like HPTLC, HPLC, gas chromatography, liquid chromatography, we can identify and we can easily make the separation of all these components. I know this procedure are already happened in our various pharmaceutical industry, but we have to make a standard procedure also in the terms of chromatographic analysis. How we will segregate these pigments, how we will make a standard solvent, because in doing chromatography, we have to make the solvent system. So there should be a proper segregation. And that segregation part is going to compare with the standard one. What is the standard? The various pharmaceutical company like Merck and Sigma Aldrich. So they were preparing this standard. So when we put this standard in compare with these pigments which are present in our drug substances, so we can easily correlate and we can easily compare with that. Okay, so that should be a very challenging job in pharmacy practice. So the segregation part always plays a major role. Clear? The last one is the time integration. While doing preparation, the time management plays a major role. Clear? Suppose when we talk about trituration, so the minimum time is 1 hour, 60 minutes. So it may depends on the nature of drug substances. So suppose if the drug is very hard like a stone, like a piece of stone, so that will also make a more than 1 hour. So the time integration also plays a major role while preparing this drug substances or the medicinal substances from that particular sources. Moving towards the next slide. Now these are related with the compounding and combining effect. Now what it is? The word compounding means when A combined with B and when it forms C. So that property, the physical and the chemical, the therapeutic property of the C is not resemble with the A and B. It is something far away from A and B individually. Clear? So this is compounding. Okay. Now what is combining effect? The A and B, when this A combined with B and when it forms AB, not C. So this AB resemble with the physical and the chemical property of A and B individually. So these are the effects in our pharmacy. So we should also focus on that in the terms of synergism. It is a very important terminology in the pharmacy. What is synergistic effect? So synergistic effect is the extra and ordinary effects by combining these two elements or more than two elements. So while doing the mixture, we have to make the synergism. Don't make as a compound. It should be reflecting something more effective in terms of therapeutic value, in terms of analytical value. Everything should be included in that. So these are also challenging while doing the various mixture. It also, while making this mixture, it reduces the ADR. Now what is ADR? Adverse drug reaction. So when we make mixture, when we make compounding, when we make combined effect, so that particular compound and the molecule and the drug substance and the drug particle should not have any ADR. Clear? So when it produces more ADR, more adverse drug reaction, so that would not be considered as a safer side and that would not be considered as a safe drug or safe medicine or safe remedy. Clear? So that should be also a challenging while doing the combination and the mixture. Accelerate the particle size. Always remember one thing, whenever we do preparation, whenever we do pharmaceutical practices, so we are not only talking about the drug or the whole system, we are talking about that particle size or the atom or the molecule or the ion or the nanoparticles in our ultra high dilutions, which are going to accelerate it from lower energy shell to the higher energy shell so that it can gain the energy, not lease the energy. It is just like an endothermic reaction, not exo. So these are again challenging in our pharmaceutical practices. So these are very, very important parameters. What are ADR? What is synergism? What are the types of mixture? How we will accelerate the particle size? So this will also make you as a most specific, precise and evidence-based practice. Clear? So this is a scientific, this is the logistic behind the dynamization, behind the potentization. Why we do dynamization? Why we do potentization? So why we do potentization? The answer is to accelerate the mass particle into the massless particle from lower energy level to the higher energy level, so that it can work or plays a major role on the dynamic system. That is the answer in two lines, not in detail. So this should be included in our pharmacy practice. Moving towards next slide. As we know, this is a unique feature and specialization in our homeopathic pharmacy, that is potentization. What it is? So potentization means it is a mathematico-mechanical process to arouse the latent curative power of the drug substance. Clear? So what is the uniqueness behind that? Again, the potentization make a mass substance into massless, and that will also converting into the isotopic molecules, and that will make a collaboration or integration with the water molecules with some memories of medicine, and these are going to work on the dynamic plane. So this is actual potentization. We are doing trituration, we are doing succussion, but what is the logic behind that? The logic is like this: to make it as a more feasible or the permeable in the terms of dynamization. Now, what are the challenges? We have to make a specific design. We have to make a specific method while doing potentization. How we can make it as a unique? By introducing or by inclusion of the photonic effect. In somewhere, while doing the trituration, when we do the inclusion of light, the monochromatic light on that particular part, so that will also make it as a acceleration of that particle from lower to higher energy shell. So these are something, a new method, a new phenomena, a new specific technique for making it as a more evidential and more scientific. So to introducing the photonic effect, photonic means the light, to accelerate the photons. These are the energy carrier particles. What are the photons? These are the energy carrier particles. Clear? So that should be also included in our practice while doing this pharmacy. And the rituals and workshop, again, it is very important part. While doing this scientificity, when we included our philosophical background, when we included this workshop and the rituals background in our all this preparation part, so we can make it as a uniqueness, we can make it as a complete picture in our standardization. So these are the various headings and the parameters included in terms of potentization. Moving towards the next slide. HPT. This is also known as a homeopathic pathogenetic trial. So what it is? It is again a systematic procedure to know about the therapeutic value of that particular drug substances on living entity. So this is the HPT, homeopathic pathogenetic trial. So what are the challenges? The challenges are in terms of preclinical stage, in terms of toxicity analysis, and as per the CCRH, the new guidelines for drug proving. So what are the new guidelines? Drug proving should be RCT control, randomized control trial. It should be double blind. It should be in the parallel design, because this parallel design is a part and the category of RCT. So these are the new guidelines given by CCRH and various other international platform also or international bodies regarding with this particular HPT. Clear? So while doing HPT, we have to make more focus on the phase 0. What are the phase 0? These are in vitro study, these are animal study, which we can say the in vivo study. When we talk about homeopathy, so we always focus on the healthy human beings. We are not doing or proving on animals. Very rare. So why not? When we do proving from phase 0 on animals, on bacteria, so we have to know about the toxicity at that particular concentration of drug. This is the main logic behind that. Why we do animal study? Why we do in vitro study? Because we have symptoms of mental, physical, particular on healthy human being. So why we do animal and bacterial study? The reason is that when we do bacterial, when we do in vivo as a animal as a whole, so we are going to analyze the toxicity of that particular drug, because we don't know the toxicity or the ADR of our that particular compound or that particular particle which is present in that particular drug substance. So that should be also analyzed at certain concentration. Because suppose when we talk about any medicine, Rauwolfia serpentina, excellent result in hypertension. At how many concentration? That is important. Clear? At how much concentration it may cause ADR. So that is important. Okay. Some patient took 10 drops in half cup of water of same this Rauwolfia serpentina mother tincture. Some patient took 20 drops in half cup of water. So where is the standard? In the I am talking about the concentration. I am not talking about as a drug as a whole. So where is the concentration? Where is the specific dosage? We always talk about is susceptibility. Okay. So yes, it is accepted in terms of human trial. But what about this in animals, in bacteria? At how many microns our homeopathic medicine make pharmacological changes or structural deviation or functional deviation in that particular cell? That we have to know. This make you as evidence based. Otherwise, our pathy is just like it is not completely accepted on international ground. I am talking about internationally, not nationally. So that we have to do. Clear? So always discover or do your drug proving from phase 0 to phase 4, 5, 6. So that will make a more significant value. Moving towards the next slide. Now, this is related with the molecular analysis. What is molecular analysis? So it is the computational method that will that make focusing on forecasting the molecular structure, the physiochemical attribute, and the simulation of that particle size or the atom or the molecule. So this is molecular analysis. Clear? So when we talk about computational pharmacy, so it is regarding with the structure of molecule. So when we talk about mother tincture, because it is not possible in the ultra high dilution, because our homeopathic medicines are highly diluted or potentized. So when we talk about mother tincture, so definitely, yes, the answer is to analyze the molecules or the atoms in that particular background of medicine. Clear? So this is molecular analysis. So again, the computational pharmaceutics, it also facilitated or it is also forwarded by AI tools, by various data. So this AI, what is AI? Artificial intelligence. So with the help of this, we can make a molecular analysis. We can make the analysis of that chemical composition, that chemical compound, that particle in that particular drug substance. So that will make a drug delivery system in more effective, cost effective, and data driven. So these are the molecular analysis. So we have to also focus on that particular part while doing pharmaceutical practice or while doing pharmacy as a whole. Moving towards the next slide. PK and PD model. pharmacodynamic and pharmacokinetic. What are these? So these are the branch of pharmacology. What are pharmacodynamic? What the drug does with body. What the drug does with body of that particular living entity. Either it may be plants, bacteria, or human beings. So what is pharmacokinetics? What the body does with the drug. A reverse action. Okay, the response of body and that particular cell and the genetic material against the drug. So in pharmacodynamic, there are various actions like stimulation, inhibition, excretion, toxicity, okay, and idiopathic also. So these are all pharmacodynamic action. What about pharmacokinetic? These are related with the absorption, distribution, metabolism, excretion, and toxicology. So these are the parameters in pharmacokinetic. So while doing this animal study, or somewhere what we can say proving of our animals or therapeutic value on a specific species of animal. So this will give you an idea how the aldehyde eluted particle or that particular medicine travels in the various channel of that particular whole living organism. So this is pharmacodynamic action. How this drug travel and give their response on that particular target cell or tissue. Now, what is pharmacokinetic? When stool, urine from that particular to the pharmacokinetics. So this is again a challenging job in our homeopathy. Moving towards the next slide. Now it is regarding with the medical chemistry and the pharmacognosy. So how this will help you as a more challenging in pharmacy, which deals with the complete knowledge of drug, with the intersection of chemistry, with the pharmacy, by making a specific design and developing the various pharmaceutical drugs. So when we talk about pharmacognosy, so we have to make a complete picture of drug from their identification, from their selection, from their collection, from the preservation. Not only preserving the drug, we have to preserve the alkaloidal property, the chemical composition, the chemical particles, and the drug particles in terms of therapeutic value. So this is pharmacognosy. So we have to do this as a holistic approach. We have to do this as a complete picture. So this is a medical chemistry and it is regarding with the pharmacognosy. So again, it is very, very challenging and it is very, very important for the medical science, how we study and identify the drug, how we deal with the drug particle, how we make a mixture, how we will prepare the medicine from the drug, how we will make the standardization, how we will make the qualitative analysis, how we will make the quantitative analysis. Suppose if there is one example, again chromatography. So in this technique, the both possibilities are given, quantitative aspect and the qualitative aspect. Quantitative aspect means to segregate the n number of pigments which are present in that particular drug. What are the qualitative aspect? To qualify and to make it as a more effective in terms of their intensity at different or the certain wavelength. So these are the various aspects in same chromatography. So this will give you an idea how to evaluate the drug, how to make a standard drug, how to make the qualitative analysis, how to qualify or quantify our particle size. When we qualify and quantify this particle size, then they can be easily go to that particular target cell at that particular intensity, at that particular wavelength, so that they can show their effect. So these are the major, what we can say, background behind this pharmacognostic study. Why we have to do pharmacognosy? So this is regarding with that particular point. Now the next is moving towards the next slide. Advancement in homeopathic pharmacy. So what are the advancement in homeopathic pharmacy? The advancement in terms of research, the advancement in terms of the development, the advancement in terms of the formulation, the advancement in terms of the distribution, the advancement in terms of the acceleration, the advancement in terms of the clinical usage of the medicine. When we talk about the BHMS first year students, so we have various practicals like short experiment and the long experiments. So what we will do, we have to make a different applications like lotion, liniment, ointment with different drug and vehicle ratio, not only by the pharmacopoeia, and we have to do the analysis of that particular external applicant. So this will make you an effective research in terms of pharmacy. What is research in pharmacy? This is a specific design and this is an advancement in terms of external applicants, in terms of lotion, liniment, ointment, various medicated cosmetic products. Now, what are the developments? When we talk about newly added sources, because now in pharmacy we have newly added sources. What are the newly added sources? The newly added sources are isotes, allersotes, tautopathy, which is also known as a synthetic drugs. So we have to evaluate a new sources for the development, for the making a new medicine, a new remedy. So development should be always in terms of the evaluation, evolution of the new sources. Formulation, when we talk about various preparation, so there should be a different formulation, solid, liquid, semi-solid. So these are in all the ratios, drug vehicle ratios. So formulation always done in the terms of miscibility, in terms of transparency. Suppose when we mix A, B, C, D, and when it forms C, but in C, there is no miscibility. So this is not an advancement. It is just a mixture. When we do combination of A with B, with C, with D, and when it forms C, and this C is miscible with the component of A, B, C, D individually, then this is the advanced. So we have to make more patents. It is very glad to share this. In our Parul University, from the Department of Homeopathic Pharmacy, we published our new patent, a new design towards a sanitizer, towards an external applicant. So this is a utility of our intellectual property in advancement of pharmacy practice. This is our outcome. It is not needed to always talk about the theoretical part. We have to also apply it over this. So this is an advancement. What is the distribution? It means the PK model. Okay. How our ultra high diluted medicines travel in the nervous system, in the peripheral, in the circulatory system, or in terms of dynamization. So this is just like a great challenge in our homeopathic fraternity. When we do research like that, then we can make our pathy as a evidence-based practice. Only the, what we can say, like modern pharmacology and modern pathy, it is evidence-based medicine. Why? Because they are talking about our drug substance, our medicine in all these terms, in terms of pharmaceutics, in terms of pharmacology, in terms of pharmacognosy, in terms of pharmacodynamics, in terms of pharmacokinetics. Where is homeopathy? The homeopathy should be like that. So it is not only the part of first year syllabus. It should be extended up to fourth year. There should be also B Pharma in homeopathic pharmacy. There should be also like M Pharma in homeopathic pharmacy. PhD in homeopathic pharmacy as a separate section. So this will make you a strong academic and the independent, what we can say, a very strong influencing part in our homeopathy. And when we do this, so we make a different models. We can analyze this as an individual. So this will create a huge picture regarding with the pharmacy. That pharmacy is not only regarding with a few textbook. It is not only regarding with the few pharmacopoeia. It is a huge sea. So we can always have this multi-dimensional spectrum in our intellectual property. Moving towards the next slide. Now it is regarding with the pharmaco information. Now, what is pharmaco information? This pharmaco information is regarding with the drug discovery. How we will discover our drug particle, how we will identifying our drug particle, how we will quantify and qualify our drug particle, how we will accelerate our drug particle in the living entity, how we will make our drug particle at the higher energy level, how we will make our drug particle from mass to massless. So these all are the various parameters in drug discovery. Drug discovery, it is not only like to discover and to finding out new sources. It is regarding with all this aspect. When we do this in a channel, in a integration with the multidisciplinary, then we can make our pathy as a holistic approach. Second is integration of multiple scientific technologies. Suppose when we talk about AI tool, when we talk about the machinery technology, so these all should be integrated in our practice while doing pharmacy. So that will give you an information regarding with the drug. So that is also a very challenging job in our homeopathic pharmacy. By including the chemistry, by including the biology, by including the pharmacology, by including the pharmaceutical techniques, by including the various extensive AI and the information technique, we can make it as a best specific precise practice in homeopathic pharmacy. So this is all regarding with the pharmacoinformatics. So that should be also carried out from one generation to another generation. Pharmacy, when we talk about BHMS, so pharmacy definition is like identifying, selection, collection, preservation, preparation, potentization, drug proving, drug dispensing. But when we talk about PG students, so pharmacy is that branch of science which make a complete knowledge regarding with any source, with any drug in terms of their physical property, in terms of their chemical property, in terms of their pharmacognosy, in terms of their taxonomical classification, in terms of their chemical composition, in terms of their biochemical composition, in terms of their pharmaceutics, or in terms of their therapeutic value. So this is an advancement of our knowledge from BHMS to MD. Now, when we talk about PhD, so it is something very far away. How we can make a standardized method, how we can make a specific design. So this is a PhD. This is an outcome of our PhD. PhD is not just like to make a copy-paste system. Clear? So it is to finding and evaluate our intellectual property in a right direction to make our pathy as a evidence-based practice by giving a complete knowledge of particle size which are present in our drug or medicine or remedy. So this is pharmacoinformatics. Now this is a last one. There are some other challenges, but during this hour and this time interval, these are the major ones. Pharmacogenomics. What is pharmacogenomics? Drug substance through which our genetic material, our genomes are going to respond. Suppose when we talk about constitutional medicine, deep acting medicine. So what are these? These are the medicines, these are the remedies these are the drug substances which make their effect, their pharmacological action up to the genes, up to that particular genome, up to that particular genetic material. So this is a separate branch of pharmacy. So it is known as a pharmacogenomics. So when we talk about pulsatilla, when we talk about nux vomica, so suppose when they are the polycrest medicine, their effects seen on the various system of human beings or any living entity. So how this pulsatilla particle, not only pulsatilla as a whole, this pulsatilla particle from tincture to dynamic plane, it accelerated and give their effects and make the physiological, the morphological changes in that particular genetic material is an important evidence. So materia medica should be like that. It should not like only the collection and the preparation and the data of that particular drug. Materia medica should also teach us like a various pathway, various design, various channels through which our drug particle accelerated from one place to another place, from one shell to another shell. So this is again an important part. So these are the various skills, these are the various aspects through which we can evaluate our pharmacy in terms of evidence-based practice, in terms of scientific medicine, in terms of more logistic approach, so that we can do. Suppose if we have X drug and a Y drug, so first we have to evaluate their historical background, their taxonomical classification, then we evaluate their chemical composition. After evaluating this chemical composition, we have to segregate these chemical parts as a quantitative analysis, and along with the quantitative analysis, we have to also do the qualitative analysis by various tools of standardization. Afterward, we have to make a segregation of this active part along with this complete part of drug and make it as a comparative study on the drug proving. So when we do like that, so we can make a holistic approach. We can make a scientificity. We can make a more strong bonding in our drug, medicine, and remedy. So these all are related with the pharmacogenomics. There are certain aspects. Suppose when we talk about patent, so patent, there are multiple options. You can also patent your design. You can also patent your idea. You can also patent your methodology. There are various aspects and the branches of this patent. So always remember one thing. When we discover any new formulation, any new external applicant, so that would make a synergistic approach. Clear? So this make or this will be very effective in terms of efficacy, in terms of therapeutic value, in terms of pharmacological action. So these are the various evidence-based practices. These are the various challenges. These are the various advancements in our homeopathic pharmacy. So today we discuss about various parts, various factors, various components, various parameters for making our pharmacy as a evidence. Now this is our last part, and next time we will discuss some more models, some more experimentation, some more researches which make our pharmacy as a holistic. Thank you.
Thank you, Dr. SURAJ Bhadoria. It's really a very excellent session, and you really show the importance and relevance of the pharmacodynamics and how homeopathic doctor need to do all these things by means of evidence, and what are the things possible, how one can manage all these things. These are the things when we are able to do all these things, definitely homeopathy will be at the top of the world. Congrats for such an excellent brainstorming presentation from you, Doctor.
Thank you,
Right.
sir.
Since only 4 minutes remaining, I invite Dr. Hariharan sir to say a few words. Sir, please unmute your co-host now.
Doctor, your class is very informative. Congratulations. Doctor, I like to ask one question that in our, I know certain doctors are mixing these potencies and giving medicine, and they argue that they are getting result. So I don't know, I am not using like that, but certain doctors are doing, I know certain doctors are doing likewise. And how that will happen? And do you have, your institution have any research regarding that, doctor?
Yes, sir. For this question, we have one research like Allium Cepa lotion in the simplex and the complex variety. In the complex variety, this Allium Cepa mixes with some other medicine. So the answer of this question is when we talk about the mixture. So suppose when this component of A drug combined with the B drug with some other C drug in the same potency in that particular media, so there is a synergistic effect. So that will make a more efficacy and that will make a more effective in terms of their therapeutic value. So the important thing is when we do this mixture, this A drug should be complement with the B, with the C. This A drug should not be antidote with the B and C. This A drug should be follow well by this A, this B and C. So when we do mixture like that, so we can make a synergism. But if suppose this A is antidote or inimical with B, like Rustox and Apis, so by mixing and making mixture with Rustox and Apis mellifica, it is not going to show their synergistic effect. Why this medicine are inimical? Because the phytoconstituent of this Rustox and the active principle or the phytoconstituent of this Apis mellifica, they are not going to accelerate together at a particular intensity or a particular wavelength or the particular media. So that's why these are the inimical. So always remember one thing, whenever we do mixture, so what these people are doing, they are doing mixture with the more than one compound A, B, C. And when it shows synergism, so definitely the answer is yes, it is more effective. But suppose if these are the inimical or antidotal, so they are not going synergism. Somewhere they are producing the ADR. So these are the justification of this type of practices.
Very good, very good, very good. Doctor, see, one of the defect is that we cannot find out which medicine is acted.
Sir, our time is already over. There is a big session coming in our Malayalam session. So we need to conclude the session.
Okay, doctor. Okay, doctor. I have a lot of questions to you, but
Definitely.
lack of time, I am withdrawing. Okay. Anyway, it is a wonderful, wonderful subject.
You can say vote of thanks to the session also, sir.
Okay, on the behalf of IFPH office bearers and see a lot of doctors are hearing this, for them also, I thanking you. Your class is nice. Come again, doctor. Okay, good night.
Thank you, sir. Thank you so much.
Okay.
Thank you, Dr. Hariharan sir and Dr. SURAJ SINGH BHADORIA for such an excellent session in this evening. And tomorrow we will have a session from Dr. Venugopal Reddy. We will be talking about the horror humor therapy. That is something new for us. And Dr. Venugopal, he's a first year doctor from USA, will be talking. So hope all of you will be joined the same time tomorrow. You can use the same link also for the session. Thank you all. Good night.
Thank you, sir.
Good night. Good night.
It's over to Dr. P. Shaila to moderate the Malayalam session.
Okay.